We'll understand glycolysis in a very easy way. As this topic seems difficult, it is very easy. We'll make it easy according to exam point of view, like GATE XL and GATE BT. So what is glycolysis? We'll go through this process step by step. And where is the glycolysis site? Glycolysis is basically conversion of glucose into pyruvate molecule, and this takes place inside the cells or inside the cell. And where it takes place? Inside the cells. It takes place inside the cytoplasm. So basically glycolysis is the conversion of glucose molecule into pyruvate molecules. And the first steps involve conversion of glucose into glucose 6-phosphate. It is catalyzed by the enzyme hexokinase, where ATP is utilized. As you can see, phosphate is added in the glucose. So this is the enzyme hexokinase which catalyzes this reaction. Now, in the next step, this glucose 6-phosphate will be converted into fructose 6-phosphate by the enzyme phosphoglucoisomerase. As the name indicates, the glucose is isomerizes into fructose with the help of this enzyme. In the next step, so this is the enzyme 3D structure. Now in the next step, phosphorylation of fructose 6-phosphate takes place with the help of enzyme phosphofructokinase 1. So this is the rate limiting step. Why this is rate limiting step? Because here glycolysis is committed to make pyruvate, and this is inhibited by ATP citrate and low pH, and activated by AMP and fructose 2,6-bisphosphate. Now in the next step, we can see this is the structure of the phosphofructokinase 1. So in the next step, fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyetone acetone phosphate with the help of the enzyme aldolase. Now simple way to understand how fructose 1,6-bisphosphate changes to glyceraldehyde 3-phosphate and dihydroxyetone acetone phosphate by split arrangement by the aldol enzyme. So this is, you can see in the figure. Now there is conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Here is the addition of the phosphate group which lead to conversion of it into 1,3-bisphosphoglycerate. Now phosphoglycerate kinase, there is formation of the first ATP and there is conversion of 1,3-BPG into 3-PG. So there is addition of the inorganic phosphate to ATP to form ATP. In the next step, there is conversion of 3-phosphoglycerate into 2-phosphoglycerate with the help of the enzyme phosphoglycerate mutase. And in the next step, there is enolase enzyme is involved here. Enolase enzyme works basically by removing water molecule, and it converts 2-phosphoglycerate into phosphoenol pyruvate, and there is formation of one water molecule. So in the next step, phosphoenol pyruvate is converted into pyruvate, and here also one ATP is formed with the help of enzyme pyruvate kinase. ADP combines with the inorganic phosphate to form ATP, and phosphoenol pyruvate is converted into pyruvate. So let us see the energy calculation of the glycolysis. So here in the first step, there is a requirement of an ATP. In the third step also, second step also, ATP is required, not third step. And there is formation of NADH, and there is gain of 2 ATPs. So glycolysis energy calculation show net gain of 2 ATP and 2 NADH from one glucose molecule despite using 2 ATP initially and generating 4 ATP later. So thank you. Hope this video helps you. If you like the video, please subscribe to channel and like my video, and I am happy to answer any of the queries